Hello everyone, welcome you all to the EPG Pathshala Botany. Myself Dr. Vidya-mitra, working as an assistant professor in the Department of Botany, Savitribai Phule, Pune University, Pune. Today I will talk on jasmonic acid. Jasmonic acid is a plant hormone. Chemically, it is a derivative of cyclopentanone and it is involved in various cellular processes in plant. Interestingly, it is also playing an important role in plants' biotic and abiotic defense responses. In this module, we will study how jasmonic acid is synthesized, conjugated, transported and inactivated. We will also see the physiological effects that this hormone has on plants and the molecular mechanism involved in jasmonic acid signaling. First, we will see how jasmonic acid is synthesized in plants. It is synthesized by the octadecanoid pathway. Its biosynthesis starts in the chloroplast. First, activation of lipase releases fatty acids from the membrane lipids and free linolenic acid is served as the precursor for jasmonic acid biosynthesis. Linolenic acid is further oxygenated by a specific lipoxygenase to form 13-hydroperoxy octadecatrienoic acid. This is abbreviated as 13-HPOT. The 13-HPOT is subsequently cyclized to yield 12-oxophytodienoic acid, that is OPDA, by the enzymes allyl oxide synthase and allyl oxide cyclase respectively. After this, OPDA is transported from chloroplast to the peroxisome and its movement is facilitated by the ABC transporters. In the peroxisome, the olefinic bond of the cyclopentanone ring is reduced by the enzyme OPDA reductase and NADPH is used as a cofactor. After being reduced, it undergoes 3 cycles of beta oxidation by the acyl-CoA oxidase 1 enzymes, multifunctional protein and L3-ketoacyl-CoA thiolase and converted to jasmonic acid. Now, we will see after being exported from the peroxisome to cytosol how the jasmonic acid is further metabolized. It is either conjugated with amino acids, glucose or esterified to its methylated form methyl jasmonate in the cytosol. Among all these JA conjugates, JA isoleucine is the most active form and plays a significant role in orchestrating plant defense responses after injury and insect herbivory. The esterified form of jasmonic acid, that is methyl jasmonate, was first detected in the essential oil from the petals of Jasminum grandiflorum. This esterification process is catalyzed by the enzyme jasmonate O-methyl transferase. Methyl jasmonate is extremely volatile in nature and easily diffusible through the membrane. Therefore, it is considered as an important cue for both intra as well as interplant signaling. Catabolism of jasmonates includes the conversion of jasmonic acid and jasmonic acid isoleucine to their respective 12 hydroxy derivatives, that is 12-OH-JA and 12-OH-JA isoleucine respectively. Cytochrome P450 are well known for hydroxylation of small molecules and inactivation of phytohormones. Two sheep members CYP94B3 and CYP94C1 are involved in two successive oxidation process of JA isoleucine. Now, the question is, are JA and its various forms mobile? After wounding or herbivory, jasmonic acid level is increased within 2 to 5 minutes throughout the plant. Therefore, jasmonates are known as long distance mobile signal. Wound induced JA is transported to the root and is responsible for the systemic increase of JA level in the root. Methyl jasmonate is also mobile and it moves through both xylem and phloem. If methyl jasmonate is applied exogenously, it first diffused in the leaf tissue and entered the phloem stream along with photoassimilates. However, due to exchange of constituents between xylem and phloem, methyl jasmonate also enters the xylem stream mainly to the source via photoassimilates. Now, we will see the physiological effects of jasmonates on plant. Jasmonates modulate the expression of a variety of genes and play major role in plant photosynthesis, growth, senescence, germination, reproductive development and defense response to biotic and abiotic stress. Jasmonates decrease the expression of genes that regulate photosynthesis. RUBPKase and RUBPKase activase are the two major photosynthetic enzymes down-regulated after jasmonate treatment, hence photosynthesis. Gibberellic acids are the growth promoting hormones that play important role in stem elongation, leaf expansion, etc. To promote growth, bioactive GA binds to the cognate receptor and leads to proteasome dependent degradation of its repressor protein DELA. However, JA inhibits the degradation of DELA and restricts growth. In addition to photosynthesis and growth, jasmonates also regulate various genes involved in leaf senescence. Exogenous application of jasmonates causes chlorosis that may lead to senescence. Moreover, it is known that jasmonates interact with many kinases and transcription factors involved in regulation of leaf senescence. Jasmonic acid repression of the photosynthetic protein RCA or Rubisco activase is responsible for leaf senescence in Arabidopsis. Jasmonates also play an important role in germination and seedling development. However, it was later known that the intermediate of jasmonate biosynthetic pathway OPDA is the compound inhibiting the seed germination. Jasmonates are important for the proper development of reproductive organs and show different phenotypes. For example, jasmonate insensitive mutant of Arabidopsis thaliana showing a male sterile phenotype. On the contrary, the JA insensitive tomato mutant JAI1 is female sterile and does not develop seeds. The main intermediate of the JA biosynthesis, cis-12 oxophytodiionic acid, seems to be the essential for proper seed development. Jasmonic acid also inhibits flower bud formation. Jasmonates are accumulated after wounding and herbivory. Jasmonates are known to be the signal for induction of stress responses. Jasmonates induce the accumulation of major anti-herbivore defense metabolites and anti-nutritive proteins. For example, jasmonate induce indole glucosinolates in Brassica species. In Nicotiana, after herbivory, jasmonates are transported to the root and initiate the biosynthesis of nicotine. Another defense metabolites 17-hydroxygeranyl diterpenoid glycosides is also increased after herbivory in a jasmonic acid dependent manner. Jasmonates also induce the production of volatile organic compounds required for indirect defense. Not only defense signaling, a trade-off between plant growth and defense metabolite accumulation is also seen to be brought by herbivory-induced Jasmonate accumulation. Jasmonate-induced defenses impose significant costs on a plant growth. Jasmonates inhibits photosynthesis and plant growth. The resources are used for synthesis of defense metabolites which provide resistance to the herbivores. Now, we will see the mechanism by which Jasmonates regulate gene expression. From the previous slides, we know that the most bioactive form of Jasmonates is JA isoleucine. It is structurally and functionally similar to the phytotoxin coronatin. Coronatin insensitive protein belongs to a highly conserved F-box protein that recruits the substrate to SKP1 Col1 F-box protein, E3 ubiquitin ligase complex. Another protein called as Jasmonate Zym domain or JAS protein is a transcriptional repressor and binds to MYC transcription factor. The complex of COI1 and JAS is the receptor for JA signaling. Bioactive JA isoleucine binds to both the ligand binding pocket on COI1 and as well as the peptide of a bipartyl JAS1 degron. JAS1 degron has a conserved helix for COI1 anchoring and a loop to trap JA isoleucine in the pocket. A third essential component of this complex is inositol pentakis phosphate that interacts with both COI1 and JAS adjacent to the ligand and potentiates this complex. There is another co-repressor called as topless and the adapter protein called as novel interactor of JAS or ninja attached to this complex. The binding of JA isoleucine to the SCF COI JAS complex leads to the degradation of JAS protein by 26S proteasome pathway. Degradation of JAS releases the MYC2 transcription factor from repression and induces the expression of genes that are required for stress response. It has also been shown that the JA biosynthesis intermediate OPDA also regulates expression of J response genes in a COI dependent and independent manner. To summarize today's topic, jasmonic acid, we have seen that jasmonic acid is an oxylipin which is partly biosynthesized in the chloroplast where OPDA is formed, partly in the peroxisome where jasmonic acid is formed, and finally jasmonic acid is released to the cytosol. After it is released to the cytosol, it is further metabolites to different derivatives. Among them, jasmonic acid isoleucine is the major bioactive conjugates which plays an important role in defense signaling. Previously, it was thought that only jasmonic acid is the bioactive compound, but later on it is known that its precursor and intermediates are also playing a significant role in defense signaling. COI1 is the receptor for JA isoleucine, and JA isoleucine and COI complex binds to the JAS protein which is the repressor protein for defense response genes. Finally, with 26S proteasome, this complex gets degraded and the defense responsive genes get activated. Thank you.